Hi, everybody. I'd like to thank you for attending this webinar. My name is Simon Ramshaw, and I'm the Managing Director of Erchonia Lasers Limited, which is based in the UK. Robert's first webinar was last week, so this is sort of a follow-up to that. For those of you that know him, will know that he's a very progressive, reconstructive orthopedic podiatrist. He's widely known for thinking outside the boxes and encourages others to do the same. He's really expanded his scope of practice, and I know he's a big advocate in others doing the same. He's done quite a bit of research for Erchonia Lasers and Erchonia Corporation, and has special interest in non-thermal lasers, pain management, fat cell management, and immunology. So at this juncture, I'd like to pass you across to Rob, and he can take you to the next level. Thanks, Robert.
Thank you, Simon. Okay, guys, welcome to another lecture. We're going to look a little bit more closely at some of the stuff that we hit on last week. So what we're really looking at now is a little bit of the scientific underpinning, but primarily how the cellular process works, what we know from research, and pretty much how this research applies. So the first thing, we're going to take a whistle-stop tour of the principles really behind this whole field of photobiomodulation, which fortunately or unfortunately non-thermal lasers fit into. Then I want to do a brief look at what we think we know, and this is pretty much about cells, cellular interaction, and cellular structures, and how the laser influences those. We're going to move on from that then, and we're going to look at why non-thermal low-level laser. Again, we're going to support that with the research, and we're going to have a little closer look at what happens to our body processes during this whole aging cycle. And then with the conclusions, I'm going to allow people really to draw their own conclusions from this. I'm going to tell you my conclusions and then see what conclusions you have yourself. If you look at the screen, you will see that there's a little piece on the drop-down menu to the right-hand side where you can type in some questions. So if you have questions, please, as they come up, type them in there, and either Simon or Julie will read them out at the end. So what do we know about photobiomodulation? Photobiomodulation basically is the application of non-ionizing light to the body. And this is done in two ways. It's done in many ways, but from a medical point of view, we're primarily doing this in a couple of ways. The first way is through light-emitting diodes, and the second way very much is with non-thermal low-level lasers. And these things, as we said last week, work on the visual light spectrum, and that visual light spectrum is pretty much from 400 to 700 nanometers. Some of us can see a little on either side of that, and more of that then goes into the near infrared electromagnetic spectrum, again, as you can see there, which is between 700 and 1100 nanometers. So what is photobiomodulation? Primarily, it is a non-thermal process. It acts on our body's receptors, primarily through chromophore and chromophore interaction. And when this happens, we see photochemical changes. If we talk about an LED, a light-emitting diode, this is more like taking a blanket and putting it over the skin. And when we put the blanket over the skin, it just basically warms the top of the skin, acts with the top of the skin. So if you think of a light-emitting diode, the light from the light-emitting diode is more spread across the skin. When we look at lasers, this is more of a penetrative light. Again, both of these have beneficial outcomes, and both of them have good therapeutic indicators. But as somebody like me who works with non-thermal low-level lasers, I'm interested in what happens from a cellular point of view, and basically how we can use these lasers to improve function, remove pain, reduce inflammation, pre and post-op, and also how we can use this as an immunomodulation device in order to help us improve our own health. Other areas, of course, would be wound healing and fat cell management. Excuse me a moment. God, I think I may need some laser. I'm just going to mute myself for a moment. And of course, when you don't have laser, water is great. So as I mentioned a moment ago, we talked about the visual light spectrum. And just to recap from last week, I'm sorry, I just need to mute this a moment. So just to go back, we spoke about the visual light spectrum. So this is what we see from visual light. So we're starting around the 400 mark and going up to the 700 mark. So the basic principles that underpin this whole concept of photobiomodulation is quite simple and relatively straightforward. And basically the agreement is that the application of any form of photobiomodulation, with the exception, of course, of hot lasers, is that they don't damage or impair tissue and they don't lead to any form of tissue dysfunction. What they do, in fact, do is they lead to tissue healing and they reduce inflammation and the other things that we mentioned earlier. So this is a reference presentation. So the presentation will be available to you. And if you look at the bottom of the screen there, you can see the references that I've cited. You will find on most of those references that there is a hyperlink. So non-thermal low-level laser is also called cold laser, and it tends to remove itself from the whole area, commonly referred to as photobiomodulation, primarily to differentiate itself from light-emitting diodes. We also then have to look at the term non-thermal laser as opposed to low-level or cold lasers. The reason I use the term non-thermal is because some thermal lasers can be cold, some cold lasers can be hot. The lasers that we're talking about are always non-thermal, so they generate no heat, so we've decided to call them non-thermal low-level lasers. So primarily the target of our lasers is cytochrome C, as we said last week, which is found within the inner membrane of the mitochondria. And cytochrome C is probably a vital part when it comes to electron transport and when it comes to allowing cells to function. And it's possibly the most misunderstood one of our cells. When I was teaching and when I was learning, the mitochondria was very much referred to as the powerhouse of the cell, and that is where it stopped. But if we start looking at the mitochondria in another way, we have to ask ourselves some questions, and we have to ask ourselves, is it possible that our bodies has everything they need for a happy, healthy life, and that the only thing that we're missing is the inspiration of function? So when we look at the research around cells, taking lasers out of the picture for a few minutes, when we look at the research around cells, what do we know? So if we do a literature review of the mitochondria, and the reason it's a literature review of the mitochondria is that is the primary import port for light from a non-thermal laser. So a literature review of the mitochondria basically reveals hundreds of papers. I've kind of looked at the 6 most recent, and I'm looking at the mitochondria in aging, because from the moment you're born, you begin to age. So this is very relevant when we talk about what lasers can do. And you'll see as we work a little bit through the research, as we do a whistle-stop tour of the research, that the mitochondria is no longer something that we can view as this powerhouse of the cell. Instead of that, it's basically a platform. It's a platform that manages communication within the body, so communication and signaling within the body. It also has a responsibility within immunity and immune modulation. And something that is more coming to light now is the activity of stem cells and how the mitochondria has an interplay into those. So each of these can possibly give us clues to how the mitochondria may regulate that whole process of aging and age-related illness and disease, because all of us as clinicians, whether we are podiatrists, chiropractors, general practitioners, dietitians, it really doesn't matter. All of us are working with a cellular process, which is the product of aging. So if I have a patient coming into me with a sore knee, the chances are it's because of some exercise or some sports work that they have done, and their body isn't recovering as well as it did when they were 15. So pretty much when we start looking at the whole process of cells, we have to look at how they age. So when we look more directly at how they age, they have a very profound effect on our tissues. They allow our muscles to weaken. They allow pretty much for our hair to go gray and fall out. They allow our eyes not to function properly, and our immune function changes as a response of this aging process. What does all this have to do with lasers? It has more than you think, because if you listen to what we're talking about at the moment, you'll begin to hear where the laser will come into this. So in science, we have struggled always to explain what it is that kicks off this aging process. We look at somebody who's just born, we look at them at 13, we look at them at 18, still lots of energy, still doing fine. So what is it where does this aging process start? So when we think about this aging process, the two important figures in this at the moment are Madauer's maturation accumulation hypothesis and Kirkwood's disposable soma theory. Both of them are quite interesting. And both of them really, we could spend a whole series of lectures on them. But just to briefly go through a little bit of it, Madauer was interesting insofar as he thought that the whole process of life depended on us getting to the stage where we reproduced and we replicated, but more of that in a moment. When we look at aging again, and we look at Kirkwood, as you can see from the paper that's up there, again, this is about stomach stem cells and aging. In particular, how they change and balance tissues. Again, lots and lots and lots of papers out there about all of this. When we look about immune remodeling with age, again, it's all there. You're going to have a copy of this presentation, so you can work through this in a lot more detail than we can cover in a 35-40 minute presentation. So what I'm trying to do at this stage is just wet your whistle with what's out there, and then you can go and read the research. So when we get back to Madauer's interesting thing was the fact that we reach a certain age where the body needs to replicate. So until that time, our body's immunity, so everything that keeps us healthy, is focused about that one event. And that one event is, now it's time to reproduce. Now it's time to make a copy of ourselves. And once we had made that copy, we had fulfilled our genetic potential, and we had basically reached a stage where our bodies could go into decline because we had ensured the propagation of the species. So when you think about that, it kind of makes a little bit of sense. It kind of sounds a little bit strange. His whole thing was that our lifespan was pretty much irrelevant to the whole process of replication. And he then started talking about accumulation of dysfunction within cellular processes. What he discovered even back then was that the accumulation of damage within tissues had some interplay from the mitochondria, which he couldn't quite focus on. But his whole thing was, once we had replicated, we went into decline because basically our bodies were no longer interested in maintaining this cellular activity and this cellular function at the level it did. And as we went into decline, nature took its place. Kirkwood, who is still alive and well and in Simon's part of the world, very interesting character, because his whole theory was about somic energy, and that as we age, we have less disposable energy. So when we're young, like Madawa, he had a belief that we were geared towards this genetic replication. But he took it a stage further and explained it a little bit in a way that was more interesting. So to cut to the chase on it, what he basically was working on was, when we're young, we have buckets of energy. When we are maturing, we have buckets of energy. When we hit 20s and beyond, we still have energy, but the minute we have kids, we don't have quite so much energy. From a psychological point of view, we could say that's because they keep us awake half the night, and that's why we don't have energy. But when we look closer at the body, what we see is this somic energy that we have becomes less, and our bodies begin to focus on specifics. So when we start talking about bodies focusing on specifics, what basically happens is our body will focus on an injury, an illness, or a condition, and all the energy goes towards trying to solve that problem, trying to solve that condition, trying to heal that condition. And while our body is preoccupied with doing that, in creeps another condition, another disease. And the body isn't so much interested in that because it's got enough to cope with on what it's doing somewhere else. So when we talk about the mitochondria, we're looking at a structure which simultaneously has a lot to do with the energy of youth, but it also has a lot to do with the decline of age. So what does it have to do with the decline of age? So when we look at the research that's out there, we basically see that the mitochondria itself begins to lessen in function with age. And as we get this lessening in function, we also get this lessening in energy. So the relevance of the mitochondria from an aging perspective isn't really a new idea. It's been around for a long time. But we know that as we age, we lose the ability to produce stem cells stimulated by mitochondrial action. We know that we lose the ability to synthesize and process mitochondrial DNA, mitochondrial RNA, again, a stimulated faction of that. We also know that as we age, we become product to inflammaging, and as a result of that, we see raises in interleukins. So when we look at some of the research that's out there, especially the mitochondrial basis of aging, as you will see when you work through this later, there is a lot of very interesting stuff out there. Basically, the evidence is showing that the decline in mitochondrial function also contributes to the aging of cells and contributes to the way tissues within the body react as a result of this aging. So when we look at the whole thing of aging, what's the biggest hallmark of aging that we know? It's basically this inflammatory state, a state that's now more often being called inflammaging. And what we see when we have this inflammaging state is an increase in circulating inflammatory biomarkers, so interleukin series. We see problems with C-reactive proteins, increases there. We see very much, as I already mentioned, an enhanced age-dependent immune response. So the body has to spend too much time working on one thing because it doesn't have the disposable somic energy to work with something else. And to recap, we're seeing loss of mitochondrial DNA synthesis, so mitochondrial DNA RNA synthesis. We're seeing an impairment of stem cell process and a lot more. So again, the research is there. You will be able to click on the research links here and it will take you to the appropriate papers. So this again is just to show you that there is a lot of stuff out there, especially when it comes to immunosuppressants and stuff like that. That paper was so good I decided to put it up twice. So all of this leads us then to another question, which is the whole thing of methylation. So methylation of the D loop, is it good, is it bad? If we want to stay alive, we need it. If we are going to die, it helps us to do that. So basically this methylation cycle, it is good and it is bad if we don't get them in balance. And as you can see from what we've presented so far, as we age, a lot of this goes out of balance, and we don't see differentiation working the way it should. We don't see our body regulating itself the way that it should, and we don't see the specificity that's needed to maintain intimate structures and tissues as a result of impairment of mitochondrial action. So when we look at the whole thing of mitochondrial action, I love this slide because I always get blown away by the fact that the mitochondria, the genome, the mitochondrial genome, 16.5 kilobytes of information, of memory storage, of energy storage, that lessens with age. It doesn't need to lessen with age. Why not? Let's wait and see. This is now a slide that's giving you the breakdown of these processes. And all of these processes, I'm not going to work through them now, but when you start looking at this and work through these processes, all of these processes are influenced, controlled by, governed by, communicated with, or exercised by mitochondrial action. So why non-thermal low-level laser? We know from the research that I've presented to you that the mitochondria and the activity of the mitochondria decrease with age. That's not new. We know that this should, mistake there, this should say non-thermal because I'm using that term now. So we know that non-thermal low-level lasers increase the viable number of mitochondria. So if we increase the viable number of mitochondria, we also increase the energy of youth. We also increase the ability of that stem cell to do what it's supposed to do, or even for that stem cell to be there. We also see less change within tissues, because when a tissue changes, you can get many different changes of different nature within tissue. Again, mitochondrial action. So we know that non-thermal low-level laser has a global effect on the body. The research tells us that. And again, here is the research, and as you can see, you can link into that. So laser light, to me, I kind of look at laser light as the new drug, because our body contains, in my view, everything we need for a happy, healthy life. We have all the cells we need. We have all of the energy we need. But as we age, we lose the energy. We need a way of putting the energy back. Laser basically works just like a drug. It recognizes and reacts with molecular receptors, cytochrome C. It activates, as we said last week, that secondary cascade, biphosphate to triphosphate. It affects the behavior and function of cells, as we saw last week, upregulation of the interleukin series, expression of stem cells that don't usually get expressed as we get older. We see increases in the amount of energy. We see balancing of the interleukin series. We see cytokines, everything coming into play. We see lessening of fat function. All of this as the result of laser. So really, the body has everything that it needs. The only thing it's missing is that I suppose, inspiration to function. And we know from one of our colleagues in Russia that laser now is known to downregulate functions. When I was in college, it was a case of we can't switch anything off, we can't downregulate anything, we have to upregulate around it to create balance. So when we look at the biological effects of low-level laser, this slide tells us all of the biological effects. So if you look at the center of the slide, you see the mitochondria. You can see our waves of laser light coming into that mitochondria, and you can see what basically is happening. We're having an analgesic effect. We're having increased lymphatic flow. We're having stimulation of stem cell metabolism. We're having accelerated wound healing, pain relief. And as you can see from that, lots and lots of things that the mitochondria is doing that in the past we would have said it didn't do. So that's what we know so far. So the conclusion of this, is it possible to conclude, and I think it is based on the evidence, that non-thermal low-level laser triggers many, many, many processes through mitochondrial action. And when we trigger that process, the only limit to that process is the experience of the clinician. And when you look at Erchonia's lasers, like these lasers are used in pain management. They're used in pre and rehabilitation. They're used in neurological management. They're used in autistic studies. They're used in Parkinson's studies. They're used very much in weight management. They're used in body sculpting, diabetes and obesity management. They're used in podiatry, chiropractics, dermatology, wound care. Because the human body is that pharmacy that contains everything we need, the only thing we're missing is the inspiration to do that. These lasers supply that inspiration. If we look at it another way, in terms of every one of us has a genome, that genome is a map. Or we could say that genome is, more correctly, an index. And that index is an inventory of everything that our body is supposed to have. But as we age, we're not able to produce as much, to live as much, to enjoy as much as we used to, because we lack the energy, the inspiration. Based on what I've presented to you, we could argue that non-thermal low-level laser is that inspiration. This small, medium, large, whatever size Erchonia laser you have, is the very device you need to go to the next level. So next week, we're going to talk about all of these lasers, but just to give you a slight introduction to them, this is Verjus. Verjus is a green laser. A green laser has strong affinities with fat cells, and it has a very, very strong affinity with collagen and cellulite. When we look at Zerona, the red laser, again, primarily this was one of Erchonia's first biggies because this was used before liposuction procedures to improve the quality of the exudate that was being sucked out of the body. It worked so well that surgeons started saying, well, hang on, we don't need to do all of the sucking. We can just light them up and let their body do the rest. Then for those of us that work in podiatry, we have this device that is great for dermatophytes and molds, but it's also great for improving circulation, improving the quality of nails, so restoration, rejuvenation of nails, as well as the dermatophytes and molds. Then we have the violet laser, which is the EVRL, which is interesting because the violet laser is a little bit different because it's more like, let's see what's wrong. The violet laser helps us to establish what's wrong. It also helps us to establish and program things that are wrong in order to make them right. It has a huge affinity with the sympathetic nervous system, which, as we know, is very important. But more so than that, it creates quite a lot of reactive oxygen species and H2O2, which is great when it comes to immunity, because let's say we get the seasonal flu virus. What happens when we get a virus? We get increases in oxidative stress. And oxidative stress just basically means that within the body, we get a buildup of reactive oxygen species, we get a buildup of free nitric oxide, and the body can't clear it out. So because the body can't clear it out, making it simple, what happens is we get an increased viral load. So now let's say we take a laser like this violet laser, which creates a lot of H2O2, hydrogen peroxide. When we look at this electron transport chain, let's say we go into oxidative stress and we target that oxidative stress with this laser, we're now getting the body to produce something that it wasn't producing as a result of that viral infection from that flu. We're getting it to produce H2O2. What's that doing? Flushing all the crap out of the system. Another interesting device that was my favorite for a long time was the PL Touch. PL Touch again was used very much in pain management. It was used very much by the chiropractors in the States. It's used quite a bit in general practice. Again, a red laser. And of course, now we have the new FX635, which I use in my clinic for neurological work. So before we go, just to recap, what I've tried to present to you today is all of the things that happen as a result of aging. I've given you briefly what happens in terms of our cells in that process. And then I've told you what non-thermal low-level lasers do in terms of that process. So as we age, all of the things that we are losing are reproduced by these lasers. So if you've got a laser sitting at home, sitting on your desk in your office, take it home, shine it on you, shine it on your family, shine it on your pets, increase the energy. Do what Erchonia do, bring a little light to life. Thank you for listening. I'm gonna reduce my screen and hand you back to Julie. Or is it you, Simon, that I'm...
Well, it can be either. We're both on the call.
Okay, so if there's any questions, I'll let Julie work with that, or you can work with that.
Yeah, there is one question, if you can hear me.
Yep.
Hi, Robert, thanks for the well-researched presentation. Are there any papers looking at downregulation in situations like cancer, for example?
I think this is research that needs to be done. There aren't any papers directly looking at cancer at the moment. One of the things we have to worry about with, or we did worry about with cancer, was proliferation of cells, because when we use lasers, we are allowing the body to proliferate cells. And that's one of the reasons why we say, look, if you have a patient that has an active cancer or has had cancer in the last number of months, don't treat them. My experience of that is something different. Now, based on the research that I've presented you today, I'd ask you to do what I do quite often. Just step out of the box for a minute and think, what have we got? We have a thing called a genome. That genome is the menu, the recipe. It is the map of everything we were born with. And our body basically processes illness based on what that genome says. So if you were born with cancer, I think if you were born and you had cancer that day, then the laser would do damage to you in terms of that cancer. But if you were born without it and only had the potential to have it, the body knows that that's alien to you. So the body will try to fight that. And it can't, because it doesn't have the energy. Think about the presentation. It doesn't have the energy. I'm not saying go out there and shoot a cancer, but what I am saying basically is the body knows that it shouldn't be there. The body doesn't have the energy to sort it out. I treat cancer patients, but I treat cancer patients in terms of pain management. I'm not treating their cancer. I'm treating their pain. We have very good outcomes. I hope maybe that answers the question, but it should raise for you quite a lot of more questions. And by raising those questions for you, I think you will find the answers yourself within the presentation. Hope that answers it. If it doesn't, type another quick question and Julia will scream that at me too.
There's
Thanks
no
a
more
lot for
right
that,
now.
Rob. Any more, Julie?
Not right now. I will let you know if any come up in the meantime.
Okay.
Okay. Well, I'd like to thank Rob for that. It's very insightful. And the whole purpose of these educational lectures is to try to open up the minds of everybody, because there is so much that we can actually do here. A lot that even Erchonia haven't scratched the surface of yet. But we're going to keep pushing the boundaries. We invest over $5 million at any one given time in research and development.